So happy to talk about sunflower sea stars, what we're doing here at the Alaska Sea Life Center. So sunflower sea stars, what are we looking at? Well, these are echinoderms, so they're related to our other sea stars, urchins, sea cucumbers, those sorts of things. If we look at the scientific name, the genus, the species, Pycnopodia, literally translates into dense feet, and helianthoides literally means sunflower. So we have a sunflower sea star. These are pretty cool sea stars. They're the largest sea stars in the world. They get to be about 1 meter in diameter. For reference, this is about 1 meter high, so they get to be large. They're large sea stars. However, we really don't know a lot about them. We don't know a lot about their natural history, their role in the ecosystem, but we do know that they're generalist predators in our nearshore food webs. And so these guys are mesopredators, and when you think of a mesopredator, they're medium sized, and for a terrestrial sense, this is like a raccoon. This is like the raccoon in the sea. They eat all kinds of different things. They live in all kinds of different environments. And just like raccoons, they have interactions with people. Usually it's food related, or they're kind of getting in trouble. Anybody use shrimp pots, crab pots, long line, probably have had sunflower sea stars clogging your gear, grabbing things. So we do have interactions with them, both positive and negative. So I just wanted to point out the other sea star that's in this picture, this little one here, and you see those smaller arms, those smaller rays. Well, these sea stars have superpowers, believe it or not. They have this really cool adaptation called programmed cell death. And what programmed cell death allows them to do is if they feel or sense danger from another predator or something like that, those cells will naturally die, and that arm will fall off or crawl away, and basically they can crawl away themselves. And then they have this other superpower where they have regeneration. They can grow those arms back. This is both a blessing, and what we've come to know now, it's also a curse. So what happened? Well, back in around 2013, we started to see a decline in sea stars due to sea star wasting. These stars were literally melting. This impacted about 20 different species, all along the Pacific coast, but primarily the most heavily affected were sunflower sea stars. The causing agent really wasn't really well understood. They didn't know if it was bacterial or viral. One kind of leading working hypothesis was that the microflora that's actually on the tissues itself was being impacted by dissolved organics from runoff, those kinds of things, and then that was leading to this programmed cell death. So instead of just one arm dropping and crawling away, basically the whole animal shutting down and dying. However, just this last year, the Hakai Institute down in Canada, they found pretty much the smoking gun, where everybody was looking at histology, looking at the tissues and everything else. They weren't looking at the splomic fluid, and this bacteria, which occurs naturally, it's actually pretty prevalent in shellfish. This is being found in higher abundance. Sea stars can live with this, but the ratios, the relative things that are happening with that, what actually causes those population spikes, that's still yet to be understood, but this is actually what's happening with the sunflower sea stars and their wasting. And so what's the overall impact? Well, it's probably the largest ecosystem disaster that we really don't talk about. Over 5 billion sea stars, and these are sunflower sea stars, have been decimated all along the Pacific coast, all the way out from Aleutians down through the Baja Peninsula. In the southern region, they've pretty much been wiped out completely. Here in Alaska, we're still seeing them here and there. But one thing about Alaska, as many of you know, it's hard to actually access all the spots where these animals live. Sarah kind of alluded that to with the birds, but it's the same with sea stars. So they're not really as well understood, but the monitoring that we do have, we did see pretty strong declines. Anecdotally, right out our back door, we didn't see them for about 4 or 5 years. We've been recently seeing them more and more, but we also see years where we don't see them again. We also do not see the big giant sea stars anymore. We're seeing some smaller stars, which could be a recovery, which is a positive trend. So why care about the sunflower sea star? Well, one, they're a charismatic member of the tidal community, right? So we have patrons. So they're part of these tidal communities, part of the intertidal, the subtidal. But more and more, there's growing evidence to support what their ecological role is in these kelp forest communities. Most of us in here probably work, play, recreate, live on the water. And as you're in Resurrection Bay or Kenai Fjords, you probably look at the shorelines and you see those bandings of barnacles, mussels, kelp. Well, we know a lot about a sea star that lives there. Those are the ochre stars and how they help shape that ecosystem. Well, more and more research is happening with sunflower sea stars to understand what's happening below the waterline and then these other understory kelps. And what's happening is there's this proliferation of sea urchin barrens where basically there's no more sunflower sea stars, the urchins have moved in, and they're wiping out these kelp forest understories and upper stories. And we do know a lot about what benefits we have from kelp forest. They're essential fish habitat for forage fishes, salmon, flatfishes, and various invertebrates. And we've seen a similar story to this with another mesopredator, the sea otter, right? That's kind of the keystone species story that we've heard a lot about. Otters are removed, urchins come in, they make these urchin barrens. Well, there's more and more evidence to suggest that sunflower sea stars may have an even more significant impact on these communities than sea otters themselves. One, sunflower sea stars don't discriminate between urchin sizes. They'll eat big ones, small ones, ones that are high in caloric content, that are ready to spawn, ones that aren't. And then there's more and more research that's showing that just a sea star in the area or moving through the area will it actually deter urchins from moving in. So a lot's going on. These kelp forests are getting wiped out, the urchin barriers are increasing, and because of this, they were listed as critically endangered by the IUCN some time ago, and in response to that, there's international, federal, state, tribal, universities, expertise all got together and formed the POTA Recovery Working Group. And with this, they look at all facets of what it would take to actually have a recovering population along the Pacific coast for the sunflower sea star. With that, they made a roadmap to recovery. These cover things all over from permitting to aquaculture, and using aquaculture not necessarily for reintroduction, potentially in the future, but also using those larval sea stars to understand recruitment, natural and otherwise. What are the key factors they need? And continuing on, in 2023, NOAA has proposed a listing of the sunflower sea star as threatened or endangered. That listing was likely going to happen this year. It's probably going to get pushed to next year. And then once that happens, there's a lot of other things that are going to be coming into play. So what are we going to do? Like, how are we getting involved? Well, at the Alaska Sea Life Center, we have the opportunity to basically partner up with a lot of institutions with the SAFE program. The SAFE program is a conservation program structured within AZA. Those of you that are not familiar with AZA, AZA is the Association of Zoos and Aquariums, and they're basically kind of set the benchmark for accreditation as far as welfare for animals, safety, science, conservation, that kinds of thing. This allows us basically a working format to not only work with AZA facilities, but also work with other agencies as well. There's about 13 AZA facilities that are partnered up on this, and about close to 40 other agencies that are not AZA related. So collectively, we can use our expertise to look at what we can all contribute and how we can use each other. And so why the Sea Life Center involved? This is why. We have sunflower sea stars here. When we had sea star wasting disease basically hit our area, we weren't impacted in the building. It's kind of one of those, what is going on here, it's different than in the bay. We're an open system. We actually pump water from the bay, utilize it in our facility, utilize it in our aquariums, and it goes back out. So what's happening with our animals that's different than what's happening in the bay, this isn't unique to us. There's other facilities along the Pacific coast that saw kind of the same phenomenon, but there's also facilities that had wasting that hit their facility. This happened in landlocked facilities that are even connected to the ocean, so that's one of those other mysteries that didn't have natural seawater. They're using artificial seawater, and they were impacted. So, as part of our effort, this spring we teamed up with a few of our partners from Omaha's Henry Dorley Zoo. They have expertise in mariculture and also with cryopreservation. They actually are one of the gene banks for a lot of different threatened and endangered species in the world. The Steinhardt Aquarium, their expertise lies in mariculture, that kind of thing, and they're actually kind of centered in a position to be positioned if there were a reintroductory effort, that's one of the primary spots where they're looking to do that. And then the Sunflower Star Laboratory, well, that's what they do. They do sunflower star stuff, and that's all they focus on. They do in-house stuff, lab stuff, field work, education, outreach, that kind of thing. So we took our stars, and we first had to identify our stars. We had all these individuals. We actually have the largest population of wild sea stars in captivity. It's actually more than all the other facilities in the lower 48 combined. We have 47 Pycnopodia in our building currently. And we haven't really been collecting actively Pycnopodia for about 10 years. We just haven't needed to. Our animals have survived. The ones that we've gotten, we've actually gotten incidentally. We've collected, live rock or something like that, and next thing you know, that little Pycnopodia crawls out, and I guess you're with us now. So that's how we've gotten a lot of our animals recently. We did collect a little more for this project, just so we can get a little bit more diversity in our population. But we had to identify those individuals. Each one of those individuals individually, so we can tell them apart for later things. So we did morphometrics, counted the number of arms, that kind of thing. We took photos, pictures worth 1,000 words. Here we have a nice description that we can use for this sea star. But what I also want to point out with this imagery, we were able to share this with some of our partners. We're down at Friday Harbor Labs, Willem down there. He's actually working on AI training to be able to tell these stars individually. So if you look at the sea star anatomy, of course it's a radial symmetry, but they have one feature called a madreporite, which is kind of a landmark feature. That madreporite is used for the fluctuation of water in their body that they use for movement and other things. But they can use that as a landmark, and other features that they have on their body are these calcareous spines. And those spines, they don't change over time as far as their orientation. So if you look at it, it almost creates almost like a celestial map. So this celestial map they can use to identify a star. So we're contributing in that way to help our partners. Genetics, so not only do we have the physical morphometrics and the photos, we're also using genetics. We set up some 2 feet, don't worry, they grow back, that's that superpower they have. We sent those 2 feet off to UC Merced down in California, and they're working to put together some population genetic stuff for us so we can use those data later. Spawning, we spawned all our sea stars, we injected them all with a polypeptide hormone. It's a naturally occurring hormone that they have. It basically kind of kickstarts the reproductive process. So we give this injection, we have them separate, it kind of kicks things off a little bit. And this way we can finally figure out who's boys and who's girls. It's kind of easy to tell which ones are the boys in here. So we have males that are basically emitting sperm. We have females that are emitting eggs. We're collecting both of those. The sperm itself, those are the eggs. It's a better shot. You can see them kind of broadcasting those eggs in the water column there. We cryopreserve the sperm. Some people ask why we don't cryopreserve the eggs. It's because the eggs have too much moisture, and so when you freeze eggs that have a lot of water, the water makes them break, so it doesn't really work. This is technology and techniques that have been perfected, but what allowed us to do here is because our sample size is much higher, we could try different things. We could use different cryoprotectants to stabilize those and test those out. The other thing we did is we did fertilize eggs and we developed larvae. Once these larvae get developed, we can then go back and freeze the larvae, something that we haven't been able to do before, but because again, our sample size is higher, we were able to do some replicates of that. And then we let these larvae develop. These are some larvae. The reason why we had to let them develop is because at this stage here, they're static, they're sitting, they're not doing anything. Where at this stage here, they're actually able to swim around. This is when the blastula completely formed. They're starting to move around. And why this is important, when they get to this stage, we can actually ship them at this point. When they're in that static stage, we actually have, it's pretty cool, those like Amazon automatic stirrers, you guys ever seen those? But if you don't like to stir your batter, you can actually buy these things and just sit there and stir your egg. Anyway, we did the same thing. So we didn't have to stir them. This thing just gently stirred them as a way to go. But once they got to this point, we could ship them off, and that's what we did. We shipped off some brood stock and some larvae to some partner facilities in the lower 48 so they could have the opportunity to work with these animals. And then this is just that happy dad moment down here. That's it's had its first meal of phytoplankton down here. And then this is Shed Aquarium in Omaha. Here we go to the zoo there. So kind of some brief project outcomes. A really big part of this was the permitting again. We were able to secure export permits for Alaska to the lower 48. Most facilities in the lower 48, not a big deal, but in California, it's a lot more important and a lot more work. So we had to get an import permit for California, which we were able to do with the help of the Steinhardt facility. The imagery for AI learning, I talked a little bit about that. So we contributed to that. There's some other stuff that's developing out of that where basically it's a sea star scanner. It's kind of cool. You can just take any sea star, plop it on there, it scans it, and it can tell you exactly what star it is, and you're done. It's all the AI learning and all the imagery that we've been providing to help with that. So they're using that down in Friday Harbor. Genetic samples, we were able to get genetic samples from 45 of our stars. The 2 stars that we didn't get samples from are the little tiny ones. We just didn't feel it was appropriate to take in their tube feet because we need quite a few tube feet to make up a large enough sample size. These unique larval crosses, we had 8 of them that we were able to do. So these are 8 unique crosses. Prior to that, there's only 4 that had been done with our other partner facilities, so we were increasing those sample sizes. We banked 12 different males, their genetics. Larval cryopreservation, again, those were part of these 8 crosses, but those are basically developing those techniques so later on we can distribute those larvae and reanimate those larvae and start to grow them. And then the brood stock distribution. We sent a handful of different facilities. Most of them are for females because with the males we could send those gametes out pretty easily. But the females, we're still working on those techniques to actually send those gametes out. What we're working on now here in the near future, we have an animal care manual, part of the AZA process. We have an animal care manual that we've actually completed the first draft. We've actually gotten it now just this last week for a steering committee review. That will then go into publication hopefully early next year. That will be public access for everybody to look at. It's not just like how to raise a sea star, it's basically everything that goes into keeping the sea stars, including the larval work that we've been doing and other things as well. We have routine exams that we're planning for our sea stars, so it's not just a one-time look at the sea stars. That was great. We're going to look at them again to see if those morphometrics have changed at all. They're not born with 20 arms, they grow arms over time, so these animals are going to get more arms. Part of that as well is taking advantage of the animals that we didn't get to spawn. So we still have a handful of males that we don't have that genetic material from, as well as females that we can identify and do some larval crosses with. Program partnerships. We want to explore more program partnerships. We have a lot of great partners already with other programs. We have our coral partners. The folks, when they were here, they had the opportunity to go to APMI. Mike gave them a tour. They had some conversations about doing some things. And then increasing field monitoring opportunities. So I mentioned earlier, there's not a lot of monitoring specific to sunflower sea stars, but looking at opportunities to increase that so we can have a better understanding of the trends, similar to what John talked about with the sea lions. What are the trends actually doing, whether it's locally or across the state? And with that, these are some simple things, or I should say, more attainable things given resources that are available now, and this is more citizen science driven. Down in California, where there's not nearly as many sunflower sea stars, in fact they don't have any, they've had a couple that they've spotted, but they're doing more learning and outreach with citizen science. You know, have you seen the stars? This is the flyer that they developed, and basically you can report these things. This is the database that they're going to, this hasn't spread further north because with the number of sea stars people are going to be able to see here, it's going to inundate them with too much information. So this is in the lower 48, but this is something that we could similarly do here. In the meantime, if you're interested in contributing to citizen science, iNaturalist is a great a resource that is actually actively being monitored by the Sunflower Sea Star Lab. because that's what they do, sunflower sea star work. And then organizing some community events similar to what Steinhardt does down in California. They have a solstice sea star search. Solstice sea star search. Anyway, solstice sea star search, where there's some logistical challenges here in Alaska to do that, but we could actually probably correlate this with some of our lower tides that we have, like in the springtime or throughout the summer, where we can get the community more involved, not just in Seward, but statewide. But starting here locally, that's an easy way to do something like this. Marine is the multi-agency something network. It's out of Santa Cruz. This is what the original format for reporting sea star wasting. But with that, most people are just reporting sea stars that are wasting. We actually want to know healthy sea stars as well, so I would steer you towards iNaturalist, if possible. And then some of the stuff that our partners are doing that we're kind of keeping track of and assisting with as we can. I mentioned the AI stuff down in Friday Harbors, the San Juan Islands. They actually have released some juvenile sea stars that were produced through mariculture. These are about 3 and 4 year old sea stars, and they've been able to track them now for about 18 months. And using the AI, they have 4 or 5 of them that are actually staying within that same area. They're thriving, they're not suffering from disease, so a lot of things happening there. In Oregon and California, they're doing some cage studies looking at the impacts on urchins and whether they're eating urchins, and this is where a lot of that data's come out, where even if they remove the sea stars, the urchins aren't coming back just because the sea stars have been there. eDNA, we were supposed to have an eDNA talk later today. I was really hoping they could explain more about it, but it's essentially eDNA, animals are continuously sloughing DNA, and that DNA can be found in the environment. So you can take a water sample, analyze it, and look at what is there, and you can find if sea stars are present. In California, this is actually taking off right now as far as specific to sunflower sea stars. What they're finding is that they are really good at sloughing DNA. We're looking at trying to find out what the degradation time is for that DNA and what the accuracy is. So with the iNaturalist, this is actually one of the things that they've been able to do, is find these sightings, confirm that there's a sunflower sea star, and then start using some of the eDNA to see if there's more in the area. Similarly to what we did here in Alaska, they're looking at doing Project Canada and Project Lisboa in Portugal. Portugal actually has some of our not our sunflower sea stars, but sunflower sea stars. Creating a stud book with all the genetic material that we have. We actually know more and more about what animals are available in captivity. Creating an educational toolkit that can be used for learning and outreach programs for multiple people. And then the Echinoderm Academy 2.0 basically is an echinoderm culture workshop that we had down in Friday Harbors last year that we're looking to do again next year. So I think that's all I have. It's a lot of coffee. Thanks everybody. Sea Life Center, our learning team especially, they're working with these animals pretty much every day at our discovery pools, our husbandry teams, and our vet and science teams.